Dear students, previous class we have seen about the insect head, thorax and digestive system. In this class we will see about the insect integument or body wall. So what is insect body wall? So it is nothing but it is an ectodermal organ and it has functions of protection, absorption, transpiration, secretion, excretion and sensitivity. Today class, what are all the things we will see about the insect body wall is, first one is the basic introduction, what is insect body wall and what are all the functions of the insect body wall. The next one is the structure of the insect body wall and third one is the chemical composition of the insect body wall. So these three important aspects we will cover in this class. So first, in this brief introduction we will compare the human being skin and the insect integument. So this insect integument, it is nothing but a supportive structure as endoskeleton in vertebrates. So it resembles the human skin but differs in the exoskeleton and they do not have bones. Body wall pigmentation also persists like that of human being skin color, but insects have too many pigments which shows different colors like melanin, which is responsible for yellow, brown and black color, and homochromes, which is the pigments responsible for red, yellow and brown. So insects, they are having so many pigments and they are also differ from human being skin in changing the color by controlling the expression of pigments. For example, Diastus hercules shows yellow green color or black color based on the atmospheric moisture. So it also known as camouflage. So it is a different big topic. So now we will see only the insect body wall. So the insect body wall is otherwise called as exoskeleton, which is formed by plates or sclerites. The flexible lines of the integument between sclerites are called as sutures and movable points of contact are termed as articulations. It has different structure and functions. Some of the internal organs like alimentary canal, the tracheal system, genital duct and ducts of various dermal glands have integument lining which are invagination of exoskeleton. The integument gives more area for muscle attachment. The flexible lines or elastic regions of integument provide exchange of gases by ventilatory movements. So generally it is a protective structure for internal organs and prevents the pathogen entry, mechanical damage and radiation effect. Since the integument is an impermeable one, so it prevents the water loss. It also take part in insect shape and coloration. So it is a stratified structure. Stratified structure is nothing but, so it is arranged in layer by layer. So which can be divided into three basic parts. The first one is cellular epidermis and non-cellular basement membrane and the cuticle. So generally the basement membrane is the underlying layer. Above this epidermis is present and the cuticle is the outer layer. So now we will see one by one, what is cuticle, what is epidermis, what is the basement membrane, what is the structure of these three and what are the functions they are having. So the first one is the cuticle. It is the outer layer of the integument and it has three layers: epicuticle, exocuticle and endocuticle. So here the exo and endocuticle are collectively called as procuticle. The mesocuticle are also present as a fourth layer and it may be found in some insects. So the rigidity and flexibility of the cuticle varies in different parts of the body and in different life forms. So in this cuticle, the first one is the epicuticle and it is a multi-layer and it also has 4 layers. So I already told that, so integument is a stratified structure. So it has different layers. So one by one they are formed. So the epicuticle, it is a multi-layer and it also has 4 layers. The first one is the inner protein epicuticle, outer cuticle layer, wax layer and the cement layer. Okay. So, the thickness of the epicuticle is 1 to 4 micrometer. So, here the important point also, so the layers are different in thickness. So, here the epicuticle thickness is 1 to 4 micrometer. So, epicuticle, so I already told that it has the 4 layers, isn't it? So, the first one is the inner epicuticle. So, it is the thickest layer of the epicuticle and the thickness is 0.5 to 2 micrometer in thickness. It contains tanned lipoproteins because of the phenol oxidases, which are extracellular enzymes produced during damaging of epicuticle. So, the next one is the outer epicuticle. So, it is a thin layer. So, inner epicuticle is a thickest layer, the outer epicuticle is a thinner layer and it also known as trilaminar layer. So, what is the function of this layer? So, during the new cuticle formation, it is formed prior to formation of other layers and it protects the procuticle from the action of molting fluid. It is formed by cuticle or sclerotin and it consists of highly polymerized lipid. So, the next layer is wax layer. So, this is also very important layer. Why? Because we will see. So, it is a superficial layer and it contains different components like lipids and hydrocarbons. So, it is important. Why? Because the hydrocarbons. So, this hydrocarbon is responsible for attraction of the insect. For example, it is mainly present in ant. So, we can see in our homes also the ant, they go one by one. So, because of the hydrocarbon only. So, here the hydrocarbon number atoms is 12 to 50 and it may be odd or even number. Aliphatic alcohols may also be present in some insects, for example, larval Lepidoptera and Coleoptera. So, the wax layer is another important function is it is impermeable to water and also it can recognize pheromones, sex attractants and molting stimulants. Wax molecules are transported through the pore canals which are 100 Armstrong in diameter to the wax layer from the epidermal cells. In bees, the epidermal glands of 4th to 7th abdominal segments secrete the wax and it is pressed in between the overlapping sterna. This is molded by forelegs and mandibles to produce the honeycomb. So, the next layer is cement layer. It is secreted by dermal glands and protective barrier for other layers. It mainly consists of mucopolysaccharides and may be hydrophobic or hydrophilic. It is not present in all the insect. The next one is the procuticle. It is a layer of endocuticle and exocuticle. Endocuticle is not a pigmented one and it has successive light and dark layers. Vertical striations of these layers are used to estimate the age like that of our growth ring of tree. So, we can estimate the age of insect by using the vertical striations of these layers. So, these vertical striations are formed by arrangement of microfibrils of chitin on the protein matrix in a definite direction or in a changing angle. These vertical striations are nothing but pore canals which are run through the endocuticle to the outer surface of integument. So, the pore canals are 1 micrometer in diameter and several thousand to million per meter square of integument. These are all the connecting tubes between cellular and cuticular layers and transport the lipids from the epidermal cells to the surface of cuticle. Based on arrangement of microfibrils, the structure of pore canals varies like ribbon like or twisted ribbon like. So, the next one is the exocuticle. It may be thin. So, it is thin in soft bodied insect or thick. So, it is present in hard bodied insect. It is not digested by molting fluid as like endocuticle, and it is a stable layer. So, the next one is the basement membrane. It is a thin connective tissue layer or membrane of inner lining of basement membrane, and it is 0.5 millimeter or less than thickness. It contains mucopolysaccharide and non-cellular layer. It can act as an effective sieve because of its negative charge. So, it is the important function of basement membrane. It has also two layers, basal lamina which is secreted by epidermal cells, and the next one is the reticular lamina, and it contains the hemocytes and fat body cells. So, it is a continuous layers with epidermis and continuous with sarcolemma where muscles are attached. So, next one is the epidermis. Epidermis, it is a single outer layer of cells. Epidermal cells are secretory in function and secretes cuticular components and enzymes during new cuticle formation and molting. So, it is very important one. So, it is the important function of epidermal cells. So, it is secretory in function. So, what is new cuticle formation and molting? It is a separate chapter. So, how the new cuticle is formed. So, this is the basic for new cuticle formation and molting. Since they are secretory in function, they are called as gland cells. Generally, they are of three types. So, these gland cells are three types: class 1, class 2 and class 3. Class 1 cells, they produce pheromones. Class 2 cells, they are ductlets and known from the sternal glands of termites, and class 3 cells produce cement layer, defensive secretions and pheromones. So, what is gland cells? So, generally any gland cell, it contains a reservoir, reaction compartments, collecting and excretory ducts. Some of the insect gland cells are silk glands. So, you may know that. So, we wearing the silk sarees which come from silk gland of silkworm moth. So, some of the insect gland cells are silk glands, defense glands, glandular hairs, wax glands, gland cells of adhesive organs, milk glands of viviparous flies and various head glands. Other cells associated with epidermis for specific function are cells or hairs, ducts, sensilla, bristles, scales and enocytes. So, what are enocytes? They are nothing but polyploid cells with tubular endoplasmic reticulum. So, they are often associated with the fat body trophocytes and lipid metabolism. They produce hydrocarbons and transfer to epidermis through cytoplasmic strands. So, they also deplete the stored lipids during starvation. The next important one is the epidermal cell junction. So, what is the important one? The membrane organization and polarization of epidermal cells depends on cell junctions. Generally, the epidermis has three different regions or domains like apical, subapical and basolateral. They have different proteins. Apical domain has yellow protein, subapical domain has two protein complexes which are essential for junctional assembly. Adherens and septate junctions are two identical junctions in the epidermis. These are formed by transmembrane and actin binding proteins. Adherens junction is formed by E cadherin and beta catenin. It also contains nectin, aphadin for regulating the epithelial organization. Septate junction, so it is ladder like and it has cross bridges between epidermal cells with 20 nanometer gap. It contains transmembrane proteins like neurexin, contactin, neurogyllin and sodium potassium ATPase. Sodium potassium ATPase act as diffusion barrier. The scaffolding proteins present in septate junction are coracle, varicose, scribble, clitactin, disc large, sinus and megatracheae. ERT is an essential protein present in septate junction to prevent passive diffusion of molecules larger than 10 kilodalton. So, these are all the important proteins that are present in the epidermal junction. So far we have seen about the structure of the insect cuticle. So, now we will see about chemical composition of cuticle. So, what are all the components that are present in cuticle? The first important one is the chitin. So, it is a polysaccharide made up of N-acetyl glucosamine residues. So, it is in the form of crystalline microfibrils, generally alpha, beta and gamma chitin. Mostly the insect cuticle has alpha chitin which has anti-parallel orientation of sugar chains. The chitin microfibrils are 3 nanometer in diameter and 0.3 nanometer in length. About 20 chitin chains form one microfibril. The chitin microfibril are arranged both in helicoidal and orthogonal arrangement. In helicoidal arrangement, the microfibrils rotate anti-clockwise and mostly found in exocuticle. Due to this helicoidal arrangement, series of thin lamellae is formed and it is called as cuticle. In orthogonal arrangement, the layers of cuticles are very thin and their orientation differs from one layer to another layer. This is otherwise known as plywood like arrangement. So, this is about the chitin. So, chitin, what is chitin and what is the function of chitin? The next one is the proteins. Proteins are major factor for determination of properties of cuticle. So, insect cuticle proteins may be structural proteins, cuticle organizing and anchoring proteins, and they may be also cuticle modifying enzymes and proteins. They are generally 3 types. So, what are they? First one is the arthropodin, which is unturned, water soluble and non-chitinous. Second one is the sclerotin, they are tanned and water insoluble. Third one, resilin, which is a elastic protein and it facilitates the wings and legs flexibility. And third important compound is lipids. So, cuticular lipids are nothing but hydrocarbons and they found beneath the cement layer. So, hydrocarbon profiles vary with insect species and they are useful in taxonomic analysis. The last one, minerals. The insect cuticle which are exposed to mechanical stress are having metals and minerals. For examples, that in termites, mandibular cuticle has manganese and zinc. So, so far we have seen about the what are the chemical composition, that is chitin, protein and lipids and also minerals. Last one, what are the internal and external processes of body wall. Okay. So, it is nothing but like that of our nails and hairs, so which are present in human beings. So, first we will see about the internal process of body wall. So, the first one, sutures. The term suture comes from the Latin word su, that means sew, a line or narrow space separating sclerotic areas of cuticle is called as suture. So, there are 4 types of suture present in insect cuticle. External grooves that are formed by linear inflections of the cuticle and appeared as internal ridges or plates. It is for strengthening the skeletal wall and to provide surface for muscle attachment. The second one is the line of flexibility where the sclerotization is discontinuous, and third one is the lines without sclerotization, and fourth one is the true sutures or lines between sclerates. Mostly the external grooves are present in majority of the insect. And second structure is apodyme. It is an internal cuticular process of the body wall. The halo apodyme is appeared externally as a depressed line or pit. They are the multicellular invagination of epidermis and mostly in the form of ridges, plates and arms. They strengthen the exoskeleton. Tendons are the thread-like apodymes attached to the body wall. At the end of tendon, muscles are attached. Some unicellular tendons also present which give attachment to a single muscle fiber. So, they may be multicellular or unicellular. And the next structure is apophysis. So, it is nothing but a spine like internal inflection of insect cuticle. And so, first we will see about the suture. Now, the next is the articulations. The non-sclerotized region or flexible region of cuticle is known as articulation membrane or corium, which is movable. The movement depends on area of articular membrane and it is limited by one or two pair of articulation point. The one pair of articulation point is monocondylic and the two pairs of articulation point is dicondylic joint. Monocondylic joint has partial rotary movement and the hinge movement by dicondylic joint. Two types of muscle attachment with the articulation, that is intrinsic and extrinsic muscles. Intrinsic muscles are sclerotic prolongations within the articular membrane and extrinsic muscles are attached on the outside of the skeletal parts. The mouth appendages are articulate with the cranium by extrinsic muscles and the articulation of leg is by intrinsic muscles. So, the next is the external process of the body wall. The cuticle of the insect body wall has variety of microscopic structures in the form of points, pits, ridges and sculptured designs other than the larger outgrowths like spiracles, spines, hairs and scales. All the external process of cuticle is classified into two groups, that is cellular and non-cellular outgrowths. Based on the emergence of the outgrowths from the epidermal cells, they are classified into cellular and non-cellular outgrowths. The first we will see about the non-cellular processes. The non-cellular projections of the body wall are cuticular structures. They are spicules, small spines, hairs, corrugations and ridges. They are unrelated to epidermal cell and may be cytoplasmic process of epidermis. The next is cellular processes. So, they are also two types, unicellular and multicellular. Unicellular processes, they are hair-like processes and generally termed as setae. They may be of different types, namely spine-like setae, feather-like plumose hairs, flat scales, cones, pecks, hooks, spatulae and knobbed hairs. Setae are plasmatic outgrowth of single large epidermal cell. It is present inside the hair socket or setal alveolus, which is surrounded by setal membrane. The cylindrical internal cavity of setae is called as trichophore, which contains the cells associated with setae. The epidermal cells that form setae are called as trichogen and the saccade forming cells are termed as tormogen. Then the multicellular processes. So, they are generally halo outgrowth of entire body wall and are lined by a layer of epidermal cells. They may be movable, that is spur, and immovable, that is spine one. Example for spur is tibial spur and for spine is lateral claws on basic tarsus. So, these both the structures are in legs, that is tibial spur and lateral claws. Both spine and spur may have setae. Okay students, today we have seen about the insect integument. So, what are the important points we have seen? The integument is protective cover of an insect and has other important functions for the insect. It has basement membrane, epidermis and cuticle. So, cuticle is divided into epicuticle and procuticle. So, cuticular components are secreted by epidermal cells. So, inocytes are also secretory in functions for specific components. Cuticle consists chitin, protein, lipids and minerals, and it is a microfibril structure on protein matrix. Insect body wall has sclerites and plates which are united by sutures. Diversified microscopic roughenings and larger outgrowths found on the cuticle. In the next class, we will see about the insect molting.